What is mitosis? Mitosis is the way cells, which are the building blocks of life and are what all living organisms are made of, copy genetic information to make new cells in a process of asexual reproduction. It is used by cells to repair damage, growth, and development. The first step of mitosis is interphase. Interphase is where a cell spends most of its life. The cell is living, growing, and producing proteins. The white line that you see in the picture is the nuclear membrane that defines the nucleus. I made the organelles as interphase is also where the cell does all of its normal cell functions, which the organelles carry out. Now back to interphase. The DNA is completely unwound. And that is how it normally looks for most of the cell's life because they are in their chromatin form. There are proteins and DNA all tangled together. The first phase of interphase is G1, where the cell gets bigger. You can see this on the second cake to the right. This phase is where the cell makes proteins and the organelles needed for synthesis. The proteins and RNA are synthesized. The chromatids, which are the ones in the middle, are doubled, but are still considered one chromosome for each pair because they are called sister chromatids and they are connected. Centromeres are organelles that play a role in the development of spindle fibers. More on them later. This entire process of everything duplicating is called synthesis. There is one more growth phase called G2. All of this can be shown by the cell cycle model to the right. As you can see, this model is already in late G2 phase. This is the second gap that the cell grows more and reorganizes its contents for preparation. Now the cell is ready for mitosis. For the sake of simplicity, I'm just going to have 4 chromosomes, which you can see right in the middle. In reality, a cell has 46 chromosomes, but in this stage, it has 92 chromatids, which is only one side of a chromosome. This next step of mitosis is called prophase. In this stage, the chromosomes condense, making them easier to see, under a microscope of course, and easier to pull apart later on. Spindle fibers, which you can see above in yellow, are beginning to form and the nucleus has disappeared. And the membrane is preparing to break down in this stage, but it can still be seen. The next step is metaphase, where the chromosomes begin to line up, as you can see in the model, and the cells make sure that the chromosomes and spindle fibers are on the opposite sides, as the spindle fibers have a job to move the chromosomes during mitosis. The nucleus membrane is already broken down at this stage, as you can see, as there's no white line around it, which were there in the previous stages. My favorite part of the process is called anaphase, where the chromatids break apart and are pulled by the spindle fibers to opposite end of the cells because the protein glue that holds the 2 sister chromatids together is broken down. The next stage of mitosis is telophase. It is technically the last stage of mitosis, but I'm including cytokinesis, so that will be the last. In telophase, the cell is nearly done dividing and is starting to rebuild its normal structures. The 2 nuclei are beginning to form at opposite ends. The chromosomes are beginning to decondense and become stringy again. The last stage is cytokinesis, which literally means cell movement. This is the division of the cytoplasm, which is the substance which holds the cell's organelles in place, to split into 2 halves, so it can form 2 new daughter cells. I want to go for the 4, so I'm going to connect cells to vaccines, drug screening, and vitro fertilization. Cells do play a role in the production of vaccines. A vaccine is essentially a preparation that improves immunity to a particular disease. It contains a resembling disease that activates the body's immune system. White blood cells recognize, destroy, and remember this version of the pathogen. That way, the immune system can quickly recognize and destroy this disease later on. A vaccine is like a pathogen imposter. Cell culture is also important to study, as it is also proving useful in finding cell targets, or in other words, drug targets, for drug screening. The study of cells is also important in the process of vitro fertilization, as it involves the removal of cells from a female body, processing the cells in a laboratory, and reintroducing the processed cells in a process of medical therapy, and this can help with females who are suffering with infertility and do want to reproduce. That is all, folks. Thank you and good night. Hope you learned a little something. Now to eat all this cake.